before I start, I wanted to have a chat. So a lot of people, the line is beginning at office hours, which means that people are starting to study, which is great. And for those of you who intend to visit me in the next week or so in preparation for the exam, I'm going to tell you what I'm telling everyone. Draw out your structures. Add your proteins to where they go in the structures. Draw an arrow, We talked about this a little already, but people are, finding it useful. So there's your, is that right? There's your microtubule, right? So draw out your structures, and then, side binding protein X does this, and then does this. End binding protein Y does this. And not only that, but what is the outcome on the big picture? What's the big picture? What's a function of microtubules? Give me one. Someone give me one.
Transport.
Transport of vesicles. If this is mutated or this is mutated and it can't bind, how will that affect vesicular transport? Because those are the questions. If you don't know me by now, then you don't know me at all, right? You really should know me that I'm going to take it 2 steps further with everything. And so I think the problem that a lot of people are having is being able to predict what those 2 steps are going to be. And so I'm telling you right now, if you can put it all together, one big piece of paper with all the information we've learned, put it all together, and then say, if mutated, then disassembly or whatever. And then no vesicular. I'm not telling you that's what the answer is. But something like that. What's another function of microtubules? What part of cell division? What specific function? For microtubules? Splitting of the chromosomes. So it's chromosome segregation. So all of these things, if something goes wrong, what is their effect? Or if something, so just think of it, if it's overly active, what's the effect on mitosis? Chromosome segregation. If it's underactive for whatever reason, what's the effect on mitosis? Right in front. No. Actin. Right? So let's talk about actin. What's a major function of actin that we keep talking about? Cell movement. So you'll do the same thing with actin, right? Actin, monomer, and you also obviously want to have just the dimer, tubulin dimer, or an actin monomer, because things bind to monomer too, right? You'll place all the proteins where they go, and you'll assign their functions, and then you'll think, what's the effect on cell movement? What's another function of actin other than cell movement? Cell shape. Muscle fibers. So all these things that we're talking about and learning about, take it one step further. What's the role of these things in those big picture functions? And then you'll remember it. Then you'll feel organized. You'll feel like you know how to think about these things. I don't want one person coming to my office without that piece of paper where they've done that. That is the rule. Because I don't want to tell you to do this. I've told you all, even you people at your computers right now, I've told you all to do this. So do it, bring it, and I will correct it. If something's wrong, I will fix it. If you want to go over that with me, I will go over it with you. But you need to put in the work. So you've got from now until Monday, Wednesday, or Friday to put the time and effort into that. And then if you feel confident and you sit with your friend and you go over it and you feel good, don't come see me. I don't want to see your face. I got other things to do. But if you feel like you're freaking out, or not, just nervous, come see me, bring it with you, we'll go over it. Okay? Is everyone feeling good? Yes.
So just draw out all the processes, you're saying.
Like I've told you all semester, draw out your processes in some kind of way that helps you to think about them. And then you'll be able to think about them. Yes. Any other questions before we move on? Okay, good. Okay. So far, okay, so we learned about, the actin and the tubulin and intermediate filaments. So then we started talking about all these proteins that do things, that regulate cytoskeletal dynamics, regulate whether something's going to be growing, whether something's going to be shrinking, whether something's going to be on, off, whatever. And then you can put them in categories, right? We've got our proteins that nucleate by binding to the minus end. Proteins that nucleate by binding to the plus end of actin. So what's a protein that nucleates by binding to the minus end of tubulin or microtubules? Can anyone think of anything? Where are microtubules most commonly nucleated? Who's tired? Should we all take a nap? Quick nap? The centrosome. Because what's in the centrosome that nucleates microtubules at the minus end? Gamma Turk, gamma tubulin ring complex, right? Proteins that nucleate by binding the plus end of actin. What's a protein that binds to the plus end of actin and adds on monomer? Formins, right. Proteins that encourage or inhibit polymerization by binding to subunits. What I mean is monomer so either this dimer, tubulin dimer, which is a subunit that makes up microtubules, or the actin monomer. What proteins bind to monomers or subunits? Yes. Cofilin? Profilin. Profilin? Yep, profilin. Profilin's one. What's another one? That's actin, right? Staphmin. What does that bind? Tubulin subunits. And there's another one that binds. Did I see someone raising your hand? Were you raising? Did you say thymosin? It's another one. So that's not subunits. Tropomyosin binds to the filaments. So, yep, 7 subunits bound together. So that's a filament. So we're talking about one single subunit versus multiple bound together. So that's definitely an important thing to think about when you're starting to think about what the different ones do. Proteins that regulate length and behavior by severing. Who knows what severs microtubules? Who knows Japanese? Katanen. And severing of actin, gelsolin. Proteins that affect stability and organization by binding to size. There was a whole bunch, right? And then proteins that bind to filament ends, and there were a few there too. So this is a way to start organizing, because as you start to place all your proteins on your cytoskeletal components, you will be able to put them into these categories also. Okay? Okay. So today we're going to talk about proteins that are involved in filament organization, structure, and localization to different regions of the cell. And then next week, we'll probably start today, but mostly next week will be molecular motors. And then we'll have an exam again. Can you believe that? Crazy. The minute I finish grading one, I have to write another. Why are you guys doing this to me? How about we just cancel all the exams? Let's do it. And I'll just grade you based on your outfit. Yeah? Yeah. You know what that means? No Mickey Mouse sweatshirts. I'm sorry. No matter how much you like that, I have a thing against. So I was always worried that when I became a mom, like it would all go to hell. Excuse my language. Sorry, computer. It would all go downhill, and I would be like Mickey Mouse sweatshirts and bad jeans. I just want everyone out there to know that it doesn't have to be that way. It doesn't have to be that way. Okay, let's get back to work. So, in order for cytoskeletal filaments to be useful, they need to be organized and attached to a cell structure, right? Does that make sense? If you have actin just floating in the cytoplasm, is that going to do anything? Just hanging out, doing nothing. Is it gonna cause cell movement if it's not attached to the plasma membrane? No. Someone's nodding. Good. Is it gonna cause Is it gonna result in cytokinesis if it's not attached in some way to the plasma membrane? Probably not, right? If it's just hanging out, it's not going to contract anything, right? And so another example is microtubules, right? If they're not attached to the centrosome, then there's no anchoring point for anything, right? For example, if you don't have microtubules attached to your centrosome, when it duplicates to form the mitotic spindle, where will your chromosomes end up? Same place they started, right? There's no way for them to be pulled in any direction if they're not anchored to something, right? If you're standing on a dock and you have a boat attached by a rope, and you're not, if it's attached, you can pull it in. If you're not on the dock and you're sitting in the water, I mean, where are you gonna go? Anyways, so a brief, a few words. Oh, this is bad. Let's see if I can make this go down without it going through the roof. No. Okay. I don't feel like messing with it anymore. That says neurofilaments are held together by lateral contacts. So a few words about organization of intermediate filaments. They're held together by lateral contacts, and they're also held together by accessory proteins. So this here is showing. I'll turn off the light. You see a little bit better. I think that eventually I'll know which switch turns which light off. So it's just colored. So plectin is green. It's this cross-linking protein, and it's cross-linking intermediate filaments and linking them to microtubules. So you've got the blue are intermediate filaments, red are microtubules, and then there's also actin-myosin in here. And basically it creates this network. It's plectin. See how it's cross-linking. Cross-linking means it binds to one and then to the other. So this is really important. So inside the cell, you would imagine it's like this really big, ugly web, right? Yeah, maybe. And a lot of what gives things its structure and keeps things where they need to go is a lot of these cross-linking proteins. And so this starts with intermediate filaments, but we're going to go through a few others. So do you think an individual with a mutation in plectin would be viable? Who thinks yes? You think so? So if plectin is binding, is basically keeping all of your cytoskeleton together. Well, I think that they probably wouldn't be very viable, right? Because you wouldn't have any organized cytoskeletal structures. That's just my theory. Okay, so now we're going to talk about organization of actin. So I kept talking about formins and ARP and how formins are involved in formation of certain types of actin, and ARP is involved in formation of different types of actin, and how they're these gel-like networks and this and that. So part of it is due to the nucleating protein and how the nucleating protein will nucleate, right? Because ARP, it likes to bind at an angle, right? And it likes to, it creates these angled actin networks. But another part of it are the different cross-linking proteins that are involved. And then formins make these bundles, so these stress fibers. So what's she talking about? So these bundling proteins in gel, so I might say actin web or gel. Basically what I'm talking about is that ARP formed actin network that's involved in movement, usually, like formation of the leading edge on a cell. But so these bundling proteins and gel-forming proteins, they are cross-linking proteins that stabilize and maintain actin structures. So what the hell does that mean? Basically, they're binding to two different actin proteins and holding them together to create the structure that they need to be in. And so this is just an example of 1, 2, 3, 4 different actin binding proteins, and the actin binding sites are in red. So you can see that here there's it's basically a tetramer where you're going to bind actin here and you're going to bind actin here. Do you think Spectrin is going to be involved in formation of actin bundles or an actin gel or web based on where actin binds? Okay, well, we can think about it more. We'll think about it more. You'll see some pictures. The answer is bundle. So bundling proteins, they cross-link actin filaments into a parallel array. You guys know parallel, right? Parallel, right? Because your bundles, think of like a bundle of sticks. You line them up all together, a bundle. So they're going to be doing that, right? Your binding protein, so your bundling proteins. And then your web, you already have a certain amount of this going on from ARP and ARP's function and how it nucleates actin. But then you're also going to have these proteins that are going to look like that. So if you go back and you look, you can see your bundling proteins are going to be these proteins that have actin binding sites on either end, and it's just straight in the middle. And then your gel forming or web forming proteins are going to look more like this. So the bundling proteins, there are many different ones, and that's because there are many different functions of actin. So you might want to start thinking about bundling proteins and the type of functions they might be involved in. For example, you can have bundling by alpha-actinin, and you can have bundling by fibrin. And what's the main difference between alpha-actinin and fibrin? There's really one main difference, right? Anyone? Yes. Right, the length. Wasn't deeply scientific, right? This green thing is longer than this green thing, right? Does everyone agree with that? And that results in a wider contractile bundle because you have loose packing of actin that allows motor proteins such as myosin to get inside, because a motor protein is going to get inside and walk along actin, right? Yes? Does anyone believe me? Fibrin is going to pack actin filaments much more closely because it's short, right? And it's going to prevent a motor protein such as myosin from getting in there, and it's actually going to be more likely to form a parallel bundle. So these different bundling proteins are going to have different function and be present in different types of actin arrays. So looser packing by alpha-actinin allows myosin to enter, making these bundles contractile. So here's another one called Villain. Do you think it's evil? Similar in structure to fibrin, right? So it's short here, green. So there's your actin, and then these little green things are your villain. And it cross-links actin filaments in microvilli. Actually, if you overexpress villain in culture fibroblasts, so not intestinal cells, right? Where do you find microvilli? What kind of cell? Where? Like, do you find them on your finger? In your intestine, right? So let's say you just put them in fibroblasts, so a skin cell in a cell culture dish, and you overexpress villain, you'll actually get increased numbers of microvilli on those cells. So, yeah. Okay, so then there's these cross-linking proteins so there are cross-linking proteins that have the actin binding domains separated by a long linker, right? So they're, long linker. And they form these webs or gels rather than bundles, and you'll see how that looks. So the cross-linking protein, it forms a 3D structure. like so, right? So here is actin, here's actin, and it's going to bind here, and it's going to bind here. And so it forms this kind of web-like gel. You can see that looks quite different from the other ones, right? Quite different. Contractile actin, web-like gel. So this is filament. Filament promotes formation of a loose, viscous actin gel by binding to two actin filaments at right angles, and this is required for formation of lamellipodia and motility movement. You're more likely to find filament and web-like gels in cells that are moving, right? It's actually required for motility. In some cancer cells, specifically malignant melanoma, they lose expression of filament. Huh. Wait a minute. Oh, wait. Wait a minute. Something's not right here. So this is not, this, I want to get back to this because I'm not happy with what I'm seeing here. I was just looking at it a little while ago.
About losing the expression.
Because I would want them to overexpress filament. So I'll get back to you on this slide on Monday after I decide what I want to say about it and have confidence in that it's.
In the American Biomolecular, he's getting a talk on that today.
Okay, go to the talk and you let me know. Danielle will go to the talk about this and update us on Monday. Okay. Right, so actin cytoskeletal structures, they need to attach to the plasma membrane. It affects the shape, right? It affects the shape of the plasma membrane, so it makes sense that they would need to attach to it, right? So how are you going to affect the shape of the plasma membrane if you're not attached to it? You're not going to, right? If I don't, like, have my finger up against my shirt, it won't move. It's not going to just randomly start moving. Okay, so there's the ERM family of proteins, and that's ezrin, rhodixin, and myosin. They bind to the sides of actin filaments and to the cytoplasmic tail of transmembrane glycoproteins. So what does this mean? So here's your plasma membrane, here's your cytosol, and let's say you have some kind of membrane protein, a receptor, let's say, these ERM proteins will actually cross-link actin to the receptor. So what do you think this might do? What, like, when I tell you you're having receptors being bound to actin, what do you think the end result on the cell is going to be? What do you think might be happening to the cell? So this is signaling regulated, right? So normally you have this inactive form of ERM protein, and phosphorylation or PIP2 binding results in a conformational change. So it opens up into this extended form, and now it could bind to a membrane protein and actin. So what do you think might be happening here?
Like muscles, I guess, they could be moving.
Nope, and we're going to talk more about that. Muscles, that more has to do with motor proteins sliding with actin. Yeah.
Would it just be individual cell movement?
Right, individual cell movement. So, because this is a plasma membrane, you have some signaling protein. There's some activation, some signals, and it results in actin binding to the plasma membrane. So you're going to get cell movement, right? Because if you suddenly have a huge web of actin forming here, it's going to push the cell out, right? And that will form the leading edge and the first step in the movement of the cell. Does everyone understand why when you ask me if the final exam is cumulative, I say no, but yes? Because what do we have here? We've got signaling. Again, you thought you were done. No. Signaling along with cytoskeleton, right? Because it's all connected. So ERM proteins are targets of number of RTKs. So remember, when we talk about signaling, a lot of the time we're talking about increasing gene expression, but we're also sometimes talking about reorganization of cytoskeletal proteins such in order to cause the cell to move, right? Right? So if some kind of chemoattractant binds to a receptor on a neutrophil, it's going to chase it, right? How's it going to chase it? It's going to reorganize its actin so it's moving in a certain direction, right? God, you guys are killing me. Come on. Okay, everyone up. Yoga time. Yoga time. Ready? Can we do downward dog? Dr. Ehrlich did downward dog in class. It was so weird. Okay, I won't do downward dog. Are you guys following the connection between cell movement, signaling, and actin, and how it might be regulated by these ERM proteins? This is ripe for an essay question. I'm just saying. Okay. And now we move on with that little hint. Okay, so motor proteins. Okay, so we've talked about so now we're done. We're done with all these proteins that bind and regulate cytoskeletal dynamics. We're done. We're done. You can forget all about them. Wait, no. Don't forget all about them. Remember them. And now we're going to talk about motor proteins. And a motor protein is going to bind to some cytoskeletal structure and bind to some kind of cargo and move it, right? That's why it's a motor protein. So they move along cytoskeletal filaments, actin, or microtubules. They require ATP hydrolysis for the movement. Usually it's like there's two legs swinging, and that's regulated by ATP hydrolysis. There are many kinds of motor proteins. They can move along actin or microtubules, but they tend to be specific motor proteins, bind to specific cytoskeletal structures. Some carry cargo to different locations in the cell. Some cause cytoskeletal filaments to slide against each other, such as muscle contraction, ciliary beating, and cell division. What else can I tell you? Well, this is what motor proteins look like. So there's some general things that you can believe about motor proteins. They all have a motor domain, or a head region, which associates with the filament tracts. So that's going to be down here. So you can think of your microtubule or actin as a track upon which your motor protein is going to move. And it binds and hydrolyzes ATP. And then you're going to have a tail region, which is going to bind to the cargo. So the tail region is often going to determine the identity of the cargo. Proteins are going to cycle between binding, nucleotide hydrolysis, some kind of conformational change that's driven by nucleotide hydrolysis. What I mean by nucleotide hydrolysis, ATP hydrolysis. Filament release, so release from the, in this case, microtubule, another conformational change, and then binding again. And it results in this walking. And I have a nice video to show you. The motor domain determines the identity of the track. Makes sense because it binds to the track. And the tail determines the identity of the cargo, which makes sense because it binds to the cargo. So here's two motor proteins that walk along microtubules, and they're directional, and we'll talk more about it. Dynein and kinesin. Okay, any questions as we move along into motor proteins? Don't want to leave anyone in the dust. Okay. So these, this category of things that we're talking about with the motor proteins is also really nice for making lists and comparing and contrasting, because we're going to have lots of examples of things and information about how things differ. So I want you to focus on those things. Members of the myosin superfamily make up actin-based motor proteins, right? You guys all knew that, right? You think actin, you think myosin, right? You didn't really need 409 for that. But now you'll understand what's actually happening. So it's a dimer, right? Is that right? It's a dimer. There's 2 heavy chains. It's a coiled tail, so that means coil wraps around each other, so that's why it's twisted like this. And then at the end terminus is the head domain, and so there's this hinge region, and then there are these light chains, and that's what forms the head domains. So, oops, this part here, what's this part here going to bind to? Actin. And this part, what's it likely to bind to? Some kind of cargo, which is actually going to be itself in a way. Well, you'll see that in a minute. So myosin 2, which is the one that's involved in muscle contraction, bipolar thick filament, is made up of many myosin 2 molecules, and they all bind to each other. So remember, I just showed you this. Oops. I just showed you this. That's one myosin. And now they're all kind of together, twisted up together. See all these little myosin heads? Each pair of myosin heads is another myosin protein, dimer. Okay? And so they're oriented in opposite directions, and they use ATP hydrolysis to walk to the plus end of the actin filament. And so they're sliding the pairs of actin filaments past each other. So actin's actually going to be up here and down here. Okay? So in this case, the cargo is itself, right? Because it's just wound around itself to create this bipolar thick. Bipolar, meaning it's oriented in both directions. It walks in opposite directions at the same time. Thick filament, because it's thick. It's a bunch of myosins wound together. Okay, so this is what it looks like in a close-up. So you've got your myosin tail, and then there's your head. And so they're all kind of wound together, so you get this array of different heads. And they're all going to work together. Okay, so you can attach myosin heads to a glass slide and throw some actin filaments down, and it'll move the actin. You can do that in the lab. So when ATP, obviously you need to add ATP. When ATP and actin are added to the myosin heads that's immobilized to the glass slide, actin filaments begin to glide along the surface. So that says something about how they walk along. So they're moving along, but since they're immobilized, they're actually pushing the actin. They're sliding the actin. Questions? Mm-hmm. Did I see a hand? Or someone itching their nose? Okay, movie.
Myosin motors
Oh,
can be
again
attached to
with
the
the
surface of a glass slide.
volume.
Fluorescent actin filaments will bind to the motor domains of the attached myosins. When ATP is added, the myosin motors.
So this is just showing that same thing I just showed you, where you add ATP and
This
it pushes
rapid
them.
movement can be observed in a fluorescence microscope as the actin filaments appear to crawl.
And see,
across the
do you
top.
think the actin's moving? So that's all the actin that you're seeing, and it's being moved by these myosin heads that are immobilized to a glass slide. Do you guys want to see it once more? Just for fun.
Myosin motors can be
All right,
attached
so there's
to
your
the
motor
surface of
proteins on your
glass
glass slide. You add ATP, and it's going to slide these actin filaments around.
When ATP is added, the myosin motors move the actin rapid movement can be observed.
And it's
in
the actual
fluorescent
ATP
light, as the elements
hydrolysis that's
appear to
causing
fall
that
across the slide.
movement. They look like worms, huh? Okay. Okay, so there are many members of the myosin family. It's not just one, and they're part of the same family because they all contain a conserved N-terminal motor domain, and it's their C-terminus that's divergent. So they all have this motor domain. You can see right here the sequence is similar among all of these different myosins in the motor domain. But the C-terminus is completely different, as you can see here. And so that has something to do with their function, right? Because they're all going to walk along actin, so they should have a conserved motor domain because they have to all bind to actin. But the C-terminus is what determines the cargo. So when we just talked about myosin 2, it's involved in contractile activity in muscle, so it's got this long coil domain that helps it to bind to each other and create this bipolar thick filament. And then there's other myosins. There's one here which binds to actin and the plasma membrane. There's one here that's involved in vesicle and organelle transport, just to name a few. So I just want you to appreciate the fact that there's a lot of myosins. They all interact with actin because they have a conserved motor domain that interacts with actin, but they're going to do different things based on the cargo that they bind to. Okay, so here's some interesting myosin facts. Not only present in muscle, which makes sense, right? Because we've talked so much about the role of actin in things other than muscle, right? They're also found in protozoan cells and plants, but mostly found in eukaryotic cells. 9 out of 40 of human myosins are expressed in hair cells of the inner ear. Interesting. Who's taken, well, you guys are all too far advanced. There's some, oh no, that guy didn't get hired. We interviewed a guy last year who, like, his whole thing was about sound, and I never took physiology at all, any physiology. I just don't know anything about any of it. You guys could teach me better than I could teach you when it came to physiology. And he was talking about sound and hair cells, and I was just amazed. So mutations in 5 of these myosins are known to cause hereditary deafness, and the myosins are important for construction and function of stereocilia. All but one move towards the plus end of actin. So remember I said there was directionality of motor proteins. So all but one myosin walk to the plus end of actin. Now, have I told you anything about myosin walking on microtubules? No. So could you safely assume myosin does not walk along microtubules? Yes. Yes. I mean, there's always a possibility that some strange thing has happened. But okay, so here are 3 different myosins that I'll tell you about, because remember we just talked about how there are many with many different functions. So myosin 1 binds to actin in the plasma membrane and is involved in cell surface protrusions. So is mostly likely to be involved in cell movement. Myosin 2 is a contractile activity in muscle and non-muscle cells required for, it's also the same myosin required for cytokinesis, right? Does cytokinesis only happen in muscle cells? Right. And cell migration. Myosin 5 is involved in vesicle and organelle transport. And a lot of them, the functions are not even known. So how do people know there are so many different myosins if their functions aren't known? Right. And it was a really easy question. And Dylan here just answered it like it was like the most retarded question ever. But so yeah, there are genes for them, right? We have a whole human genome sequenced, and you can find a myosin by looking for the conserved motor domain, myosin motor domain. If it's a gene that has a conserved myosin motor domain, it's probably part of the myosin family of proteins, right? You can safely assume that. Okay, so let's forget about myosin for 2 seconds until you have to study. Kinesins are microtubule motor proteins, so they walk along microtubules. Many of them are involved in mitotic and meiotic spindle formation. Makes sense. One of the major functions of microtubules is to form the mitotic and meiotic spindle. Again, just like myosins, they have a conserved motor domain. They're similar in structure to myosin because they've got this globular head structure. That's what walks along the microtubule. And then they have some kind of middle coil region because they dimerize through that. And often their cargo is bound to the C-terminus. And most walk to the minus end except for one of them. Which one? Which one? I don't know. Why didn't I write that down? Don't worry about it. I'm not going to ask what moves in which direction. So do not memorize anything, as I always say. But do not memorize which direction things walk in, because I'm telling you right here, most of them do this except for this. So that just says right there, there's diversity in what these guys do. But you should memorize that myosin only works with actin. Dyneins are motor proteins that walk to the minus end of microtubules. So 2 motor proteins involved in microtubules, kinesins and dyneins. 1 motor protein involved with actin is myosin. These are the largest and fastest molecular motors. I think I have a movie. These are important for vesicle trafficking and localization of the Golgi near the cell center. And then there's this one right here, ciliary dynein. It's there's a form of ciliary dynein called axonemodynein, which drives the beating of cilia and flagella. And we'll talk a little bit more about that. So myosin and kinesin, they both share a structural core. They have these 8 central ATP binding domains, and they're similar. So they probably, when you see proteins that have similar structures and functions, you can assume they probably have a common evolutionary origin. So here's myosin, here's kinesin, and they've got this lever arm. So we're looking at the head domain. So this would be your head right here, and there's your lever arm. Here's your head and your lever arm. And it has the ATPase, ATP binding domain here. And here's where it binds to actin. Here's where it binds to microtubules. So this, strangely, is also structurally similar to RAS. What do we know about RAS function? What do we remember about RAS function? Yes. It spreads signals. And what else does it do? How is it regulated? How is it activity regulated? I heard someone say GTP, right? So GTP, it's a GTP binding protein. You guys forget so quickly. When GTP is bound, it's on. When GTP is hydrolyzed, it's off. So something similar is happening here, where when you have ATP binding and ADP binding, there's a change in the shape of the protein that results in movement. And similar in RAS, when you've got GTP binding, you get hydrolysis to GDP. You'll also have a change in shape and function of the proteins. Okay, you know what? I don't want to go into this yet because I have a much better version. So we're going to start next week with muscle contraction. But I want you guys to spend some time this weekend and the beginning of next week really trying to organize yourselves for the exam.